This video will complete our discussion of DNA replication and higher DNA organization. That's from chapter 16 of your book. So just an outline of what this video is going to cover. The majority of this video will be focused on the process of DNA replication. Then we'll move into talking about DNA repair, so what happens when mutations happen and how they're corrected. Finally, we'll discuss the DNA end problem and then go into higher order DNA structure. So by the end of this lecture, you should be able to do the following. So first, you should be able to name and describe the functions of all of the proteins involved in DNA replication. Second, you should be able to draw and label a replication bubble, identifying both the leading and lagging strands and indicating the DNA strand directionality, both the direction of synthesis and the 5' and 3' ends. Next, you also should be able to differentiate between the roles of 3 different DNA repair pathways that we'll discuss. You should be able to articulate the reason that eukaryotic chromosomes shorten during DNA replication and define the role of the telomeres in this process. And finally, you should be able to define the different levels of DNA compaction, highlighting key proteins that are required for each level of the higher order structure. So just to review, this chapter is part of a larger discussion of the central dogma of molecular biology, where DNA is used as a template for RNA, which is used as a template for protein synthesis. And remember that using DNA as a template for RNA synthesis is called transcription. Using RNA as a template for protein synthesis is called translation. And using DNA as a template for its own synthesis is replication. So remember, we're making an identical copy of the DNA. And that's going to be the focus of this video. We're going to talk about the mechanisms by which replication occur. So let's start our discussion by figuring out where replication begins. And the short answer is at a site in the DNA called the origin of replication. Generally, the origin of replication is a sequence of DNA that's really rich in A and T nucleotides, and that's because, if you recall, AT base pairs only have 2 hydrogen bonds, whereas GC base pairs have 3 hydrogen bonds, so it's easier to separate AT base pairs. It requires less energy. So here we show both replication in prokaryotes, which you should remember have circular plasmid DNA, as well as replication in eukaryotes, where the DNA is linear and is organized into chromosomes. So let's start by talking about prokaryotes. So in the prokaryotic chromosome, when the origin of replication opens, so replication begins, this creates a replication bubble that's characterized by 2 replication forks moving in opposite direction, and essentially this bubble gets wider and wider and wider until we form 2 separate plasmid DNA daughter molecules. And here you can see an electron micrograph of a plasmid that's undergoing replication. This is the replication bubble, and here are our 2 forks. And something very similar happens in eukaryotes, except in eukaryotes there are actually multiple origins of replication per chromosome. And the reason for that is just because there's so much more DNA in a eukaryote that it would take forever to replicate if we didn't have multiple origins. So here you can see where we have these multiple AT-rich origin sequences. We once again form replication bubbles, each of which has 2 replication forks that progress in opposite directions, making those bubbles get wider and wider over time until essentially they merge and you have 2 new daughter DNA molecules. And here you can see multiple origins with forks on each side that are proceeding in opposite directions in a eukaryotic chromosome. So just to highlight some key terms. So first, the replication bubble is when the two strands separate at the origin. This is the growing separated area where the new DNA is being synthesized. The replication forks you can think of as each half of the replication bubble. So the replication fork is simply where the DNA is being separated at the edges of the replication bubble, making that bubble grow larger and larger over time. So now let's actually start talking about the process of DNA replication. And it's really important to note that DNA replication requires a series of proteins, most of which are enzymes that catalyze chemical reactions. And you will be responsible for knowing the name and the function of each one of these enzymes. So here, if you look at what we're looking at, we have a replication fork. So you'll notice that the DNA is anti-parallel. So we have a 5' end and a 3' end, a 3' end and a 5' end. So we have our two parallel strands. So the first step in DNA replication is that an enzyme called helicase begins to separate the strands, breaking the bonds between the base pairs. So helicase essentially opens the replication bubble and makes that fork open further and further. As helicase is opening this replication fork, separating those strands, now we have proteins called single-strand binding proteins, and these coat the single-stranded DNA, and there are two major reasons for this. One is to protect it from intracellular enzymes called nucleases that can degrade single-stranded DNA, and the other reason is to hold that replication bubble open, because there's nothing to stop these base pairs from just snapping back together and reforming, but the single-stranded binding proteins help to keep that replication fork, to keep the two strands separated. So our next important enzyme is topoisomerase, and I think this is one of the most difficult enzymes to understand, but one of the most important enzymes in DNA replication. So the role of topoisomerase is to cut the DNA in front of the replication fork and to untwist it in order to relieve supercoiling. So you can think of supercoiling as excessive twisting of the DNA helix. So if you imagine that you had a rubber band in your hand and you were holding the two ends of the rubber band and you twisted it, then you separated the two sides of the rubber band, those twists would get tighter and tighter as you opened that bubble. So topoisomerase essentially relieves that tighter and tighter twisting that occurs on either side of the replication bubble. And it does this by actually making a cut in either one or two of the strands of the DNA, depending on what type of topoisomerase it is, and then it actually untwists it and reseals that nick in the DNA strand. So the role of topoisomerase is to relieve supercoiling in front of the replication fork. Now essentially we've opened that replication fork, we've got it open, we've relieved some of the pressure by relieving the supercoiling, but we haven't actually made any DNA yet. Well, unfortunately, we still can't make any DNA, and that's because the enzymes that actually synthesize DNA can't work from scratch. They have to have something to build off of. So our next enzyme is called primase, and it is an enzyme that lays down a short RNA primer in the 5' to 3' direction. So it's shown here in red. This RNA primer is complementary to the DNA strand, meaning that it can base pair with those DNA bases, and it lays those nucleotides down one by one, going 5' to 3'. And primase is a really important enzyme because it, unlike DNA polymerase, our next enzyme, it can perform de novo nucleic acid synthesis. So de novo is a Latin phrase that means from nothing. So essentially, it can put two nucleotides together from scratch. So primase will lay down a short RNA primer that our next enzyme will use as a starting point for DNA synthesis. So far, our important proteins are helicase, the single-strand binding proteins, topoisomerase, and primase.